Welcome to the Proteomics in Proximity podcast, where your co-hosts Dale Yuzuki, Cindy Lawley, and Sarantis Clamidus from Olink Proteomics talk about the intersection of proteomics with genomics for drug target discovery, the application of proteomics to reveal disease biomarkers, and current trends in using proteomics to unlock biological mechanisms. Here we have your hosts, Dale, Cindy, and Sarantis.
Here we are with another episode of Proteomics in Proximity. I'm your host, Dale Yuzuki, with my co-hosts,
Cindy Lawley.
And Sarantis Clamidus.
And Sarantis. And we are here today to talk about cancer, and specifically cancer immunotherapy, and looking at new biomarkers for predicting immunotherapy response. We are looking at an article published in 2021 called Plasma Proteomics Identifies Leukemia Inhibitory Factor, LIF, as a novel predictive biomarker of immune checkpoint blockade resistance. Now, this particular topic...
That is a mouthful.
It is a mouthful. Okay, the first author is Loriot. Like I mentioned, it was in Annals of Oncology, and it was published in 2021. What's interesting, right, is we're talking about immune checkpoint blockade resistance. So these are PD-1 and PD-L1 therapies that have really revolutionized cancer treatment. Specifically, I mean, backing up a bit, immune checkpoint blockade inhibitors, right? These particular ICBs called is credited with saving the life of Jimmy Carter, right? He came down with very serious melanoma. He's still alive. I think he's 98, or he's really getting up there, but his life has been saved. And this particular paper is explaining, right, new predictors of ICB resistance because it's not equally effective in other types of cancers. And go ahead.
Sorry. I just wanted to add a little bit in there. So just stepping back, one more step back is, tumors tend to evade the immune system. Right. So they tend to grow often unchecked. And one of the mechanisms for that hiding or that evasion of the immune system is this PD-1 and its ligand, PD-L1, that connection. And so these checkpoint inhibitors cut that connection and allow then the tumor in a subset of patients, not majority of patients that are treated. It's actually only, 35% effective, something around there. So yep. In a subset of patients, Jimmy Carter included, this was actually an effective way to open up the immune system to see the tumor and then infiltrate the tumor. And so some of the ways of evaluating how these ICB therapies are working is by looking at infiltration of the immune system into a tumor. Thank you.
Yeah, and for melanoma, it's extremely effective. It's for the other cancers where it's not very effective. So
it's a solid tumor. Yep. Yeah, solid tumors. Yep. Very good. Sarantis, you were just going to ask. Sarantis.
No, I mean, it's just just to mention that their antibody is a therapy based on antibodies, right? That targets either PD-1, and this in conjunction with other chemotherapies, where the success rate is getting increased. But still, as you mentioned, 35% of the patient respond to the treatment. And I think it's really important that new biomarkers or new molecules that are druggable, and they can help increase the rate and the outcome of the clinical outcome of the patient, right?
And as far as predicting,
this along goes this paper as well.
Right. And as far as predicting, right, ICB responsiveness, right, I was involved in ctDNA analysis for a couple years, right, from 2015 to 2020. And all the rage was looking at tumor mutational burden. That is looking at, right?
TMB.
Yeah, TMB. Try looking at whole exomes, right? Looking at, tissue biopsies, looking at circulating tumor DNA is what ctDNA is, and trying to get this idea, right, of, okay, if there's a high mutational load of the particular cancer, this person is more likely to respond to immune checkpoint inhibitor treatment. And they tried to normalize different enrichment methods. They tried to get, all this work around it. And it really wasn't all that predictive. I mean, the R values were about 0.7. And then a multiplex immunohistochemistry was all the rage. So I saw that wave.
So another what we call companion diagnostics. So these are tools that physicians are trying to use in order to guide an understanding of who might be better responsive to such therapies. Yeah, please go ahead, Dale.
And so companies like Akoya and other spatial, right, imaging, I think Millipore also had a 100-plex kind of thing. Anyway, there are all these different companies doing multiple particular antibodies and staining. That's what immunohistochemistry is you're staining it with antibody and detecting it with fluorescence. And they're trying to get that, and they're able to up, right, the predictive power just incrementally. So that is why the hunt for biomarkers is still on. And how do I predict?
Another point there, Dale, I'm sorry, another point also here, there's a lot of immunohistochemistry based on biopsies, right? And quite invasive.
Exactly.
Because, and then I think the most important part here is in this paper, they see the value of having a plasma proteome that's less invasive and can help and support and all the findings of things that they can see with immunohistochemistry, for example.
And the question is, right, even with the biopsy samples, even with the sort of invasive methods, it's still not predictive enough, right?
That's right. They call it imperfect predictors, right? Which, they're still seeking better predictors because still, even with those guided tools, 35% are responding and the majority are not. So if we're going to give the right treatment to the right patient at the right time, we have to have better tools for that. And can I just say that this paper is so elegantly written. I highly recommend it. I just think these authors, I don't know if you've seen them speak, both Loriot as well as Antoine Italiano, amazing speakers, both on YouTube. Highly recommend following
that's them. Italiano and Loriot. They're at the Gustave Roussy. Is that in Paris, or it's multiple institutes?
It's their
right?
cancer
Paris,
center. Grand Paris, I think, is where the clinical trials for these cohorts, the validation and discovery cohorts, are based. They're still recruiting. These are phenomenally cutting edge studies.
So we're looking at tumor mutational burden, right, from a genomics perspective. You're looking at IHC from a tissue histology-based perspective. There's even effort around microsatellite instability to go back to the genomics. Another genetic tool, right? Looking at it, again, still not good enough. And Sarantis, you mentioned, right, the power of circulating biomarkers. You might say, well, haven't people been looking at this before?
I think yes, but there was not the right tools actually, right? I mean, I think here is something to say and bless about Olink technology, right? Because gives this high throughput thing to check. Before with mass spec it was a little bit more difficult. It's not so much high throughput. I think it's more easy nowadays with having this NGS based technology and screen
and what technology? Go ahead.
Right?
And it's a cytokine, right? So cytokines are these low abundant proteins, right? So very low abundance and not easily seen with mass spec without a large amount of sample or subtraction methods. And so having the Olink PEA that hooks this proximity extension assay that hooks these low abundant proteins out of solution and helps us count their abundance is I think it's just a great use case.
And again, talking about how you close the loops, right? And you go a little bit more to the molecular. You have the LIF protein, but then brings together transition factors like STAT3, for example, right? I mean, you open up completely a pathway of target proteins that could be druggable, but also could be potential novel biomarkers. It's not like one protein, there's a completely pathway. And I think tools in the future like Olink Insight that we introduce, it will help a lot of people to understand pathways and how other actually targeted molecules, right, can help and enhance drug development processes and biomarker identification.
And Sarantis, you mentioned Olink Insight. For those who haven't heard the podcast with Hilda Anderson talking about Olink Insight, I refer to that. I think it's episode number 5. At any rate, Sarantis, there's a question that I have, right, from looking at soluble biomarkers in plasma. The question I have is this signal sufficient? I mean, somebody may have advanced, tumor status. I mean, this particular paper used advanced NSCLC as their sort of a test case. Is there enough signal there? I mean, somebody has, a tumor in a particular area of their body, but we're looking at the entire sort of plasma. I mean, the blood volume of an individual is on the order of a couple liters, right? Maybe.
That's true.
Yeah.
That's true. It's always difficult to, I mean, of course, here they have matched samples, right? We try biopsies, matched samples with plasma samples, and they try to see correlations, and then they did, as Cindy mentioned, a really elegant way to check with RNA-seq to see, for example, B cells or differentiate the other T cells, for example. I think it's looking in an omics perspective gives more power to this respect.
Yeah,
that's my feeling.
Well, and the imaging as well, right? So LIF. I mean, this. I think Sarantis, you said, the Insight tool allows you to see multiple pathways and interactions among pathways, and we expect most diseases we've seen are polygenic, and there's going to be multiple pathways that are involved, and probably a signature of maybe 20 or so proteins that we'll need to understand to really use it for, say, a companion diagnostic in the future if we move through clinical utility. In this case, it's rather remarkable that it's one cytokine that has that signal where low versus high were predictive of progression, free survival. Did I say that right? PFS, yeah, and overall survival. And that's kind of remarkable. But I also want to point out the tertiary lymphoid structure correlation. So there's actually, we've got this imaging data from these biopsies, as you say, that's very invasive, right? And that you can actually see and count these lymphoid structures, which are just sort of localized immune system, adaptive immune clusters that are responding to the tumor. And that also was a correlate to that same cytokine, which I find that to be pretty remarkable.
Absolutely. That's a great point.
So, Sarantis, why don't you take us from the top in terms of talking about the experimental design?
Yeah, I mean, they try a cohort of 95 cancer patients actually treated with immunocheckpoint blockade, anti-PD-L1, and they use the Olink Explorer 1536, like more than 1500 proteins, and among this was our cohort, their discovery cohort, and then there was a validation cohort with inflammation panel with more than 290 patients, 96 proteins, where they see actually really nice correlation with data that they have seen with the Explore panel. That's also really nice because our targeted and Explore panel approach, they go hand by hand. And that's really great and really nice to see how reproducible are our assays, even though from high throughput to low throughput, right? And that's really great and nice finding. And the first one of the first biomarkers that popping up to their data set was the LIF. That like high levels of LIF in this plasma was correlated with the poor clinical outcome of the patient treated with immunoblockade. That was the first and more remarkable actually data.
Yeah, it's a really good point, Sarantis. This is true. Like they call it discovery cohort and validation cohort, but not only is it a discovery cohort where they discovered the signal and then validated it, but they also used our discovery tool, right? The Explore. At that time, this was one of the first Explore publications. And then they moved it to a lower plex where they could get the same markers. Yeah. And of course, that's going to be a lower cost. So they were able to do more samples in that validation cohort, which I like. I hadn't thought about the fact that it parallels, the discovery and validation also parallels the intent to some extent of how groups are using our products, sometimes moving to a target 96, but sometimes even moving to a flex panel or a lower plex panel. Anyway, I like that characterization that you made.
And if I understand correctly, the discovery and validation cohorts from two independent clinical trials. Is that correct?
That's right. Exactly.
Yeah, you're right. You're right.
Both still recruiting, right? So still, I would expect more to come here.
And different type of cancer, right? Different type of solid cancer, for bladder to non-small cell lung cancer. They have different type of prostate. I think also they have different cancer types. That's like more universal, looked like more universal biomarker actually.
Yeah, I missed that. That's an interesting aspect too. Yeah. Good catch. Yeah, both out of Gustav Russi, as Dale mentioned earlier. So this was the Match R cohort, so I have it written down. I'll actually read it off. Prospective trial to study the evolution of clonal architecture of tumors from patients treated with molecular targeted agents. So that's the MATCH-R. That was the discovery cohort where they used the Explorer. And I think you mentioned, Sarantis, 90 some odd samples or patients in that were used from that. And then the premise, the validation cohort, where they used the target, our qPCR readout, it was the predictive markers of immune-related adverse events in patients treated with immune stimulatory drugs. So again, that's the premise trial. So yeah, a lot of great stuff coming out of this group.
So then, as far as Cindy, you mentioned this idea of a tumor lymphoids or tertiary lymphoid structure, right?
That's right.
And this is then a feature of good response. Is that correct?
So it's, so I believe the relationship was low LIF with tertiary lymphoid structures. So meaning, my understanding, and Sarantis, please check me on this. My understanding is when you have these sort of ectopic lymphoid organs that develop in these non-lymphoid tissues, that they're a sign of immune infiltration from adaptive immunity.
Yeah.
Okay. So that's telling us that there's some element of the tumor that's visible to the immune system, and that's like the holy grail, right? If
the goal.
If you can manage to not turn on autoimmunity in some issue, right? You don't want the immune system to go haywire and get inflammation out of control. That's what kills a lot of COVID patients, right? You want to keep that inflammation in check, but you want to open up the tumor to the immune system so you have a very targeted kill, as opposed to sort of traditional chemotherapy agents that would take, us to death's door because it killed non-specifically, would kill off all fast-growing cells, right? Which is why, people lost hair, lost nails. These are fast-growing cells in the body, and you just carry it right. I'm oversimplifying, obviously, but this is the promise of the future of cancer treatments.
Yep, and it's like these tertiary lymphoid structures are a sort of in vivo validation, right?
Yes.
That the marker is measuring what we want to measure, which is is this person responding or a person not going to respond, right?
Right. Yeah, exactly. And without having to go in and do a biopsy to see these tertiary lymphoid structures, right? If you can, as you said earlier, Sarantis, if you can evaluate it in a blood draw, that's exciting.
Yeah. I mean, you think about how sick these patients are, right? These were advanced NSCLCs was the majority of the samples. I think bladder cancer was a minority of the original 95 samples. And I think there had other tumor types in there to represent, right, the discovery phase. Sarantis, what can you mention? Oh, go ahead, Linda. Go ahead, Cindy.
You just reminded me that in the results, they actually characterize the difference in progression-free survival and overall survival. And we're talking months, right? These are people that are maybe living 2 months versus 21 months.
Yes,
and so
that's a great point.
And I think even by the time they did analysis, that, and of course you have to qualify for clinical trials. So you have to have already had a treatment fail, right? There's several checkpoints I think they have through their cancer center that Antoine talks about. But we're talking about people that are very sick and at very high risk of dying. And this can sometimes maybe give them, 5 to 10 times more time than they would have had otherwise, and hopefully with a less invasive therapy. Although I don't know as much about the side effects of this.
I mean, Cindy, you bring up a good point in that these were advanced, meaning they were metastatic, right? They already had stage 4 or stage 3, right? Really high stage lung cancer, and it already spread to other organs. And I think I was thinking about this morning, where at the 50% overall survival curve, right, it was maybe 5 months. And then with the LIF low, that's 5 times that over about 20 months. Yeah. And so I was asking my boys, taking them to school, how much is 15 months worth, right? Meaning, right, from 5 month point to 15 months more, That's the difference. And you think when every month counts, I mean, these people have, right, advanced, basically fatal lung cancer. But with this treatment, you're given a year, over a year. How much is a year of your life worth?
Yeah. And it's such a personal choice, right? Because it also depends upon what the quality of life is in that year, right? Which is something that's really,
So
as
here,
we get older, we think more and more about these things, right?
And also, I think it's a nice way to monitor, right? In the way that you can see the response and you can have something to monitor. And I think that's a big useful tool if you would like to combine other therapies in the future, right? I mean, because then you know when you monitor how people they respond, having something that is less invasive, more easy to handle in a way, more easy to understand. And I think it's a great tool, and it's a great finding, and opens a new ways of seeing science in the future, right?
Yeah, interesting. Good point. Yeah, because if it's not working, you get off it, right? And you just get that quality of life back, perhaps. But it just depends upon what the side effects of any given therapy are. And certainly the ones today are much better on overall than the ones 20 years ago.
Now, since a high level of this LIF inhibitory factor, right, leads to poor outcome, did I read this correctly? It's actually now considered a drug target?
Well,
I think, go ahead, Cindy, please go ahead.
Yeah, I actually don't know. Please, Sarantis.
I think they're thinking of, I don't know if it's somewhere in the discussion, they mentioned that they're thinking like for antibody against the LIF, right? They give monoclonal antibodies against LIF. That would be a target. Yeah, I'm guessing, yes.
Yeah.
Because I think it's like a cytokine that is like differentiation, regulates differentiation.
But
and
it
I think
depends
it's also,
on if it's, I was just going to say, it depends on if it's causal or if it's
that's the thing.
If it's the results of the disease, right?
True.
So if it's
true.
a useful thing to monitor to know how someone's responding, then we wouldn't want to drug it. But I think that's interesting, right?
You would imagine the knockout mice are in, right?
That's right. That's right.
They're in generation 2. They're in the F1.
That's right.
Got the F1 cross going on right now. So
that's right. Jackson Labs is probably
there you
all
go.
over this. Who knows?
Well, there you go. I think where it was, did they give also any clue in terms of what their next steps would be? I mean, since it's a single marker, right? Pretty straightforward to implement.
I think looks like, since the numbers they're still small, I think. But Cindy, correct me if I'm wrong there. I think it needs bigger numbers, right, and bigger cohorts and different clinical trials. I don't know, Cindy, correct me if I'm wrong. You are the expert of the big samples and the big cohorts.
Yeah, I mean, what was a 95 in the discovery, and then 292 in the sort of validation. I mean, in cancer, naturally, the numbers tend to be relatively small, right? In terms of they're not in the thousands, they're in the hundreds. Cindy, do you think that these numbers need to be larger?
Oh, for sure. Yeah. Absolutely. I think, no doubt at all. It's but I think the burden of this, and this is a question, the burden of use of this as a companion diagnostic, which I think is the immediate proposal. I think further down the line, establishing causality rather than consequential, that it's consequential to the state, that there must be a lower burden for establishing a biomarker as opposed to establishing a monoclonal antibody therapy.
Yes,
and,
a great
and
point.
I don't have that, set out in data, but I think that's the immediate or the near term exciting promise here is that it could be used as a companion diagnostic. And they actually broke down the use of LIF high versus low and the TPS scores, right, which were the ones that Dale mentioned. So the tumor mutation burden, along with, basically trying to characterize this ligand expression, the tumor mutation burden, the microsatellite instability, and the immunohistochemistry, using those which are established scores in conjunction with LIF, showed utility, right? So I thought that was a logical progression in their analysis that I thought was interesting, but also requires larger sample sizes, because now you're breaking your numbers into quadrants and you need sufficient sample numbers in each of those quadrants, right?
Yeah, interesting. Well, any other final comments before we wrap this one up?
Go
ahead, Cindy. Go ahead.
I'm so sorry. I just am so excited about this paper, Sarantis. I think I've stepped on you 3 times. I just highly recommend that people keep an eye on these authors because, like I said, they are elegant writers. They speak with such clarity, and they have been driving the use of genomic tools in demonstrating utility in cancer for years, right? Cutting edge, right on that cusp. So sorry, Sarantis, your turn.
I think the method for me it's the most important thing. Go from invasive to non-invasive methods. That's the clever thing on this story. Yeah. And of course, I mean, you can back it up with multiomics like imaging, RNA-seq. But I think the method is brilliant. That's pretty much how I see it.
All right. Well, thank you for joining us today. Till next time, take care.
Thank you very much. Thank you.
Bye-bye.
Bye-bye.
Thank you for listening to the Proteomics in Proximity podcast, brought to you by Olink Proteomics. To contact the hosts or for further information, simply email info@olink.com.